Good afternoon, everyone. Yeah, so today I'm going to talk about GABA-A receptor variants, epilepsy mechanism, and therapeutic, especially like, for phenylbutyrate. I cannot move on. It's not moving. Yeah, you have a magic hands. Or maybe I should facing. It's not advancing. Yeah.
Is it?
Oh, finally working?
We have batteries. The laser's still working. So. Okay. He's working on it back there. Yeah.
Well, it's not working. Well, can you advance for me? Or if you can do that, that'll be fine. Is it working right now?
All right.
Okay. So this back, okay, that's
We're good?
on the just a minute, okay? Let me see if you can do that. Okay, so working. Yeah, so I have a brief outlines. First, I'm going to give an introduction about our previous work. As I said, we have worked over 100 mutations in GABA-A receptor, about 7 mouse models. So I have a lot to talk, but I'll give a very brief introduction. We also talk a little bit about new GABA-A receptor mutation and then the effect for phenylbutyrate. And lastly, I'm going to talk about the gene therapy. So we have done overexpression of gamma-2 in a Dravet mouse model like about 10 years ago. So we know GABA-A receptor is a major target for anti-seizure drug development. In clinic, a lot of drugs we use the benzo-derived, a lot of them directly work against the GABA-A receptor. So as I mentioned, connectively, we have studied about 100 mutations in GABA-A receptors. A lot of them, in alpha-1, beta-3, and gamma-2. And the clinical phenotype is very wide. So that just go back to history. About 20 years ago, this is the very first case about a mutation in GABA-A receptor. I remember my mentor, Dr. McDonald, actually wrote a commentary about that. Non-waiting is over. Because originally we think GABA-A receptor is a major etiology for epilepsy, but no mutation has been identified. But this is the first one. This is by a colleague in Australia. It's a childhood absence epilepsy. So we wonder, so why this mutation can cause epilepsy? How this mutation function? Probably, as the previous speaker talked about, they can have a defect in GABA-A receptor function. They have a GABA binding, channel gating, kinetics, probably multiple factors. So this is a mutation recorded in our lab. So compared with the wild type, the mutant one, R82Q, you have a much more reduced current. But if you enlarge the current, actually the kinetics is very similar. And I did a comparison with a free assembled receptor. We said GABA-A receptor has to be a pentamer at the cell membrane. With that the subunit freely form receptor, or we can tether them as ACID did that. So you can put them together and force them to go to membrane. So we actually, in both cases, we see in both mutant heterozygous and homozygous, you have reduced the current amplitude. A single channel is also done by a colleague, Matt Bianchi, compared with the wild type. The mutant one have a normal single channel conductance if they can traffic the membrane. So the problem is they don't have enough at the cell membrane. So this is the very first surface biotination done by myself about 20 years ago. Actually 2004. Tell you how many years I've been working on these things. So compared with the wild type, both heterozygous and homozygous condition have reduced gamma-2 surface expression. Suggests that you don't have enough functional receptor on cell membrane. This in HEC cells. And this is a confocal. We use the FM464 dye to mark cell membrane. And I use ER to mark the ER, and I will do co-localization compared with the wild type. Both mutant, heterozygous, and homozygous, you have a reduced membrane presence, suggests reduced functional receptor on cell membrane. as I said, we have studied over 100 mutations collectively, and this mutation can be in alpha, beta, gamma. For alpha, you have alpha 1, 2, 3, 4, 5, 6. Beta, you have 1, 2, 3. So gamma means gamma 2. So I try to correlate for the surface receptor expression and the epilepsy severity, because when we look at all these mutations, I think the common pattern is you lose surface protein expression. So very similar to what Nathan and a colleague presented. If you have loss of function mutation in gamma 2, you're likely to have fibro seizures or childhood absence epilepsy. But if you have maybe some missense, maybe more like gaps. But for Dravet, there are few mutations in gamma 2 also associated with Dravet syndrome. Later on, I'm going to have data for mouse model. So this is done actually when Sarah was in lab. So by the way, I want to say, Sarah now, she's doing a great job in leading this symposium. She actually is a granddaughter of Dr. McDonough. So because I'm a trainee of Dr. McDonough, he's looking for GABA-A receptor. So Sarah is my graduate student. So this one, Sarah was in lab. We did for truncation gamma 2 subunit. You can see, compared with the wild type, you have a very variable expression of a mutation. But this is not useful. All this protein is stuck inside the cells. It's like junk. And we did protein modeling, found compared with the wild type, the mutant is very likely to form aggregates, very hydrophobic. And they are stuck inside the ER. The very concept that we have identified a drug based on that. And then we also did a postures. So basically, you transfect the subunit alone or receptor, you label the protein, and then you chase down, see how quickly they get degraded. And Tom actually asked a question. So we found out the wild type subunit take maybe 96 minutes half-life, but the mutant one associated with Dravet loss of function take over 4 hours. So that means instead of quickly being recycled or degraded, the mutant one just stuck there, and that probably is not a good idea inside the cells. And in the live cells, we just did a confocal compare with the wild type. You see either in HEC cells, Cas7 cells in neuron, the mutant receptor is very different, the morphology. So the wild type, you have a very smooth distribution of receptor, but the mutant one is just stuck inside the cells. And we did a mouse model. So that mouse is associated with gaps and then Dravet with loss of function mutation. And we found, compared with the wild type, the mutant protein accumulates a lot. And also across different age, like 1 week, 2 months, 1 year old, and a different brain region. Like we look at cortex, thalamus, cerebellum, you actually see much highly ubiquitinated protein. So just the protein have trouble to be removed efficiently. And also we see like age-dependent effect. And this is a recording from brain slides, like some motor sensory region, layer 6, pyramidal neuron. They receive input from interneuron. Compared with the wild type, we did a 2 mouse model. One is both are loss of function, but the one with bad protein, one is without. So the one with bad protein is much more reduced GABAergic miniature, as Eva just saw in like F3 to 5L mutation. You very reduced GABAergic miniature. But for the knockout, you lose function, but don't have a better protein, you have a much better current. So, we did a genetic epilepsy. We also want to see, because in human patients, a lot of them are not genetic. So wonder what we can learn from those acquired epilepsy. And actually, when you look at the literature, a lot of them indicate reduced GABAergic neurotransmission is the pathophysiology. This just shows some example from like a rat animal model. If you give pilocarpine, with time, you see reduced probability positive label interneurons. And the human data is very limited. But when you look at the human data, they show maybe only increase alpha 2 subunit, but beta 3 and gamma 2 are reduced. So suggest probably there are some change the receptor subunit composition on cell membrane. So it's a little bit more complicated. as said, we look at genetic epilepsy. We want to see how this function and the surface expression correlate with disease severity. And we use the gamma-2 as an example, and we showed before. So you have more receptor on membrane, maybe more fiber, just the fiber seizures. Maybe you have some mutant protein stuck inside the cells, and maybe you're more likely to gaps, but you have a lot of stuck inside the cells, and have a much more reduced surface receptor. It's a much more severe phenotype. This is what we proposed. Yeah, I showed that. So we're really unified for using this. And CURE GABA-A asked us to study some gain function variants. So we're in the middle of doing that. We are making very good progress and show some data. So we care very much about how much receptor at the cell membrane. I said because they must be at the cell membrane, and they can have function. Otherwise, not useful, as Ting Wei pointed out. So also, as the previous speaker, Paul, talked about, you have a different receptor composition. You have alpha, beta, gamma. When you have a pre-assembly environment, you give alpha, beta, gamma. They can form alpha, beta receptor. They can form alpha, beta, gamma receptor. For alpha, beta, we did a flow cytometry when Dr. Lagrange was, actively doing experiment. We found you can be 3 alpha, 2 beta, and 2 beta, and 3 and 2 alpha and 3 beta. So that can be a little bit different. But if at synapse, it must be 2 alpha, 2 beta, and 1 gamma. And so right now in lab, we are actively running a lot of experiments. So brilliant technician Debbie and also senior scientist Wang Zhen. So we have done a lot of work. So we now, instead of look at the individual cells, and now we look at the population. We look at 10,000 cells to see, we get that to see what's the surface expression, what's the total expression. And also we use alpha, beta, gamma, 1:1:1 ratio, the free assembly, and we look at the mutant receptor alone. We look at the mixed as heterozygous, which is patient condition. Basically, you give a half-half for wild type versus mutant. And compare with the wild type, and here I just show a few mutations we studied. So you can tell, compare with the wild type, the mutant have reduced surface expression. So this is not final, we're still running, but as the surface expression with flow cytometry, is the flow 3 alpha mutation is reduced. And we also look at beta, because I said you can be 3 alpha, 2 beta, or like 2 alpha, 3 beta, and 2 alpha, 2 beta, 1 gamma. So beta 3 is a little bit variable. That can be maybe how they assembled. And we also look at, this is the surface expression. Total is consistent. They are reduced. And we can, and because with the flow cytometry, we can look at the ratio of how much wild type, how much mutant in a heterozygous condition. What's the ratio? And so this is what we did. And also, we wonder, for patient, can Ravicti be useful for them? That's the most important question. And if it's loss of function, it's very easy, could be very useful. But if gain function, probably maybe you should be careful. And also, that's why we check the alpha, beta, that's in the heterozygous condition. We tagged a copy, the wild type copy with HA, and then the mutant copy we don't tag. And we can see, compared with the vehicle treated, the PBA treated, you have an increase. You have an increase like the wild type allele in the heterozygous condition. So that's good news. That means, because in patient, you will have a wild type copy, you have a mutant copy, so PBA can bring the wild type, the functional one up. And Eva just talked about the F325L. We also look at this one. We did not look as in detail as you do, but we use it as a control. We definitely notice this mutation to be different. Yeah, so we look at other alpha subunit, and with the PBA treatment, we see pretty good upregulation. But for this mutant that allele, so right now I don't have conclusive data for this one, but I think maybe after a few weeks we can have more data for that. Another graduate student, Gigi, also work on this one because we want to be very careful. We ask 3 different researchers in lab, blind. We do look at the same mutation, see if we get consistent data. So the Gigi did an experiment with biochemistry. He did Western. Compare with the wild type and PBA treated or non-treated. We can see across these different mutations, reduced total expression actually is pretty clear. But for F325L, it's not that clear. But overall, you will see with PBA, it can increase the expression. We have a quantification for that. So basically, PBA will bring up the total alpha subunit. And then PBA, as I said, GABA-A receptor, they cannot function. They must function as a pentamer. So alpha, beta, gamma, they have to be formed to functional receptor. So we also look at the partnering, beta and gamma. So here we look at the gamma 2. So the trend is very similar as alpha. So you see this mutation will have a reduced gamma, except F325L. Yeah, so this is quantification. So, we know that's really important for us to get the field, is a loss function or gain function. How do you define, how do you define gain function, how do you define loss function? This is something very basic. As Eva said, it's probably multiple factors we should consider. It's not just like a GABA binding. Plus, we know, like, in patient, you have 2 copies, one wild type, one mutant. So Monica asked me to talk a little bit about the gene therapy. We have not done that, like with like a vector. But 10 years ago, we already thought of that. We did overexpression of HA tagged gamma 2 in a Dravet syndrome mouse model with loss of function gamma 2. So we did that. So basically, in the patient condition, you have one wild type copy, a mutant one. So now we give a good one. We just simply give a good copy. So this is the mutation we studied, okay? So in the gene therapy, and we see rescue protein, and then also good news is not just the rescue gamma, the rescue alpha, and the beta 2 and beta 3. So that means if you give a gene therapy for one subunit, likely you bring up the whole team, the receptor. So that's very good news. And we did all this E-phase recording, and they can rescue. Oscillation, like GABAergic miniature, also stood up. When you give a PTZ induced, that's if you give a gene therapy, pretty much back to normal. So that's very good. Suggest gene therapy will be a final cure. So basically, a treatment option, we identified for phenylbutyrate, but gene therapy is the next step. Yeah. So basically, that's the concept. How can we bring up a more functional receptor? Either gene therapy or PBA, you give more functional receptor. That's any question? Yeah. Yeah.